My name is Spirithula Xenofantos, and my project is using naturally forming compounds to remediate oxidative stress, fertility, motility, and ASH neuron function in a Huntington's model of C. elegans. Figure 3 exhibits the model organism I plan on using, Caenorhabditis elegans. C. elegans are microscopic roundworms, also known as nematodes. C. elegans are typically self-fertilizing hermaphrodites or male worms that have a short lifespan, up to 3 to 5 days, from when they are an egg to when they become an adult. The worms reproduce very quickly, which also makes them an optimal model organism. The structure of C. elegans primarily are comprised of a pharynx, a soft cuticle, a large intestine, and many neurons like the ASH and ASC neurons. When ASH neurons deteriorate, the effects are very similar to when neurons die in HD patients. C. elegans can be injected with a solution in the soft cuticle to make the worms exhibit similar qualities and characteristics of different diseases. Figure 4 shows a worm undergoing a genetic mutation that allows it to exhibit characteristics of Huntington's disease. HD results from the repetition of cytosine, adenine, guanine, CAG protein. Normally, the CAG segment is repeated 10 to 35 times within the gene, but in HD patients, the CAG segment is repeated 36 to more than 120 times. An increase in the size of the CAG segment leads to the production of an abnormally long version of the Huntington protein. The abnormally long protein is the HTT mutation, and this protein is cut into smaller toxic fragments that bind together and accumulate in neurons, disrupting the normal function of these cells. The dysfunction and eventual death of neurons in certain areas of the brain underlie the signs and symptoms of Huntington's disease. Now, when the mutation is occurring in humans and C. elegans, the rate of oxidative stress increases exponentially. Now, what is oxidative stress? Good question. Figure 5, 6, and 7 are all naturally forming compounds that I plan to use to counteract the effects of the HD strain. Figure 5 shows ginkgo biloba, which is a plant in which the leaves can be extracted, and this extract is potent with antioxidants. Figure 6 shows EGCG, which is a catechin present in green tea and is also plentiful with antioxidants. Figure 7 shows solidracide, which is a root plant that has an abundance of antioxidants and neuroprotective properties. All of these chemicals have extensive amounts of antioxidants. This is important because the worms are undergoing oxidative stress, so they have an imbalance in oxidants and antioxidants, as previously discussed. So by applying antioxidants, a temporary equilibrium could be restored in the cells of the worms, remediating the effects of the HD strain. Zhao et al. Hypothesized that solidracide would protect the C. elegans from the harmful effects of oxidative stress and polyQ aggregation. They used the strain HA759, which expresses the HTT-Q150, a polyQ tract that weakened ASH neurons, leading to ASH neuronal death. They ran a paraquat assay, which was when 75 microliters of paraquat was exposed to the worm to induce them into oxidative stress. They used GFP analysis as an indicator for the survival of ASH neurons because they wanted to test the neuroprotective properties of solidracide. In Figure 10a, you can see that only 28% of ASH neurons survived in transgenic nematodes that were not treated with solidracide. When the worms were treated with 200 millimolar of solidracide, the neuronal survival rate increased to 41%, suggesting that solidracide is capable of remediating the polyQ neuronal death. 10b shows the different concentrations and that 200 millimolar had the lowest percent of ASH neuronal death. Cao et al. Studied the application of ginkgo biloba extract and ginseng on the rate of sarcopenia on a C. elegans model. Sarcopenia is the loss of skeletal muscles, which is a neurodegenerative disease, similar to HD in that both diseases result in the loss of neuronal cells. They were able to run a fluorescence microscopy assay by embedding the live worms on 2% agarose pads, and GFP expression was conducted using the fluorescent microscope. The results show that PD4251 worms that were exposed to ginkgo had a longer lifespan when under extreme stress from the oxidation. They also ran a motility test where the worms were tapped with a wire and then the number of body bends were compared to the pharyngeal pumping rate using the GFP expression. There was a slight increase in the number of body bends performed with the transgenic worms that were exposed to ginkgo biloba, suggesting that the ginkgo biloba did have a positive application. Zhang et al. Studied the effect of EGCG on ROS production on C. elegans using N2 and CL2070 worms. The worms were exposed to 300 millimolar EGCG for an hour in preparation for the ROS assay. The worms were exposed to juglone to induce oxidative stress. Similar to Cao et al., Zhang et al. Used GFP expression to examine the effects of the stress-induced worms on a microscopic level. The transgenic worms exposed to EGCG under normal conditions showed a decrease in ROS production, which could be suggested by EGCG's antioxidation properties, restoring equilibrium to the cell. The study is to test the antioxidation and neuroprotective properties of EGCG, saligercide, and ginkgo biloba on Huntington's model of C. elegans. The purpose of the study is to test the antioxidation and neuroprotective properties of EGCG, saligercide, and ginkgo biloba on Huntington's model of C. elegans.
With chemotaxis agar, or NGM. Mark the underside of the plates into 4 equal quadrants. Next, mark a circle with a 0.5 cm radius around the origin. Then, mark points in the opposite quadrants with a T for test or a C for control in terms. If there are too many worms, increase the volume of S-basal solution, and if there are too few worms, spin them down and resuspend them in a lower volume. Set the worm tube in a fume hood. Once prepared, the worms are good to use in the chemotaxis assay for up to an hour. First, prepare the solutions. Mix the test solution by combining equal volumes of the test compound with the anesthetic 0.5 molar azide. Next, mix a control solution of the solvent used to dilute the test compound with 0.5 molar sodium azide. Now prepare a test plate. First, pipette 2 microliters of worm solution from the pellet onto the origin of a test plate. Second, add the odorants. Pipette 2 microliters of the test solution onto the 2 T sites, and pipette 2 microliters of the control solution onto the 2 C sites. Once the worms and odorant drops have been absorbed into the agar, replace the lids, invert the plates, and allow 60 minutes for the assay time. 30-minute trial, place the worms in a 4°C refrigerator. Later, remove the plate from the refrigerator when it can be counted. Not counting them immediately and leaving the worms at room temperature may allow them to mobilize again. Record the location of worms that completely crossed out of the inner circle. Calculate the chemotaxis index. The value will range from -1 to +1, representing maximum repulsion.
Here is my budget and bibliography.